Today we're going to cover all the gram-positive cocci and fresh renewable distinction between hemolytic bacteria. So, alpha hemolytic bacteria cause partial oxidation of hemoglobin, and this partial oxidation of hemoglobin causes a greenish or brownish color without clearing around growth on the blood agar. So, there is a greenish or brownish color without clearing around growth on blood agar. This is alpha hemolysis. It includes Streptococcus pneumoniae and viridans streptococci. So, two of these strep species, Streptococcus pneumoniae and viridans streptococci. Then there's beta hemolytic bacteria that causes complete lysis of RBCs, and so there is a pale or clear area surrounding colony on blood agar, and it includes Staph aureus, Streptococcus pyogenes, which is group A strep, and Streptococcus agalacti. So there is group B strep and Listeria monocytogenes. So four of these bacteria: Staph aureus, Streptococcus pyogenes, group A strep, group B strep, and Listeria monocytogenes. And this is beta hemolysis, which is basically complete lysis of RBCs. Then we're going to study Staph aureus. It is gram-positive, beta hemolytic, catalase-positive, coagulase-positive cocci in clusters, as shown in this section. Protein A is a virulence factor that binds the Fc region on the IgG, and that inhibits complement activation and phagocytosis. So that is why it is a virulence factor, because protein A inhibits the complement activation and phagocytosis. Commonly colonizes the skin, nares, ears, axilla, and groin. Its causes are inflammatory diseases such as skin infections, organ abscesses, skin infections, organ abscesses, pneumonia, often after influenza virus infection, and also infective endocarditis, septic arthritis, and osteomyelitis. So yeah, skin infections, organ abscesses, pneumonia, infective endocarditis, septic arthritis, and osteomyelitis are all the inflammatory diseases that could be caused because of Staphylococcus aureus. And then there are toxin-mediated diseases such as toxic shock syndrome, which is very important, and it is caused by TSST1. Then there is scarlet skin syndrome, which is caused by exfoliative toxin. And then there is rapid onset poisoning caused by enterotoxins. Rapid onset food poisoning, which is caused by enterotoxins. So then there's MRSA, which is methicillin-resistant Staph aureus. It is an important cause of serious healthcare-associated and community-acquired infections. It is resistance due to altered penicillin-binding proteins conferred by MEK-A gene. It has so basically this is the variant of Staph aureus that is resistance to treatment by methicillin, and it is so because of penicillin-binding proteins. And penicillin-binding proteins are compared by MEK-A. And some strains release pantyl-valentine leukocidin, which is PVL, which kills leukocytes and causes tissue necrosis. So some strains of MRSA release panton-valentine leukocidin, which is also a virulence factor or an exotoxin that kills leukocytes and causes tissue necrosis. Now, regarding TSST1, it is a super antigen that binds to MHC2 and T cell receptors, resulting in polyclonal T cell activation and cytokine release. So it is called like a cytokine explosion or cytokine cascade. Then we have staphylococcal toxic shock syndrome, which is TSS. It includes fever, vomiting, diarrhea, rash, desquamation of the skin, shock, and organ failure. TSS results in elevated AST, elevated ALT, and elevated bilirubin. It is associated with prolonged use of vaginal tampons or nasal packing. Compared with Streptococcus pyogenes TSS, a toxic shock-like syndrome, which is associated with painful skin infection. Then we have S. aureus, food poisoning due to ingestion of preformed toxin. So it has short incubation period for 2 to 6 hours, followed by non-bloody diarrhea and emesis. Enterotoxin is heat stable, not destroyed by cooking. And if aureus makes coagulase and toxins, forms fibrin clot around itself, so that forms abscess. The important things were the causes of inflammatory diseases and toxin-mediated diseases, and then that the TSST1 binds with T cell receptor and MHC2 and causes of polyclonal T cell activation, cytokine release. Then it was the differentiation of toxic shock syndrome from that caused by Streptococcus pyogenes, which is associated with painful space in infection, but this is associated with prolonged use of vaginal tampons or needle packing and increases ASTs and ALTs and bilirubin. Also important to note was the point that a serious food poisoning is caused by ingestion of preformed toxin, and that causes a short incubation period followed by non-bloody diarrhea and emesis. Next species is Staph epidermidis. It is again gram-positive, catalase positive, but coagulase negative, and it is also urease positive cocci in clusters. It is novobiocin sensitive. It does not ferment mannitol versus Staph aureus, which does. So we have to see what novobiocin is. It is an aminobutyrin antibiotic originally isolated from Streptomyces niveus that works by inhibiting the GYRB subunit of bacterial DNA gyrase, blocking DNA replication. So it is an antibiotic, and Staph epidermidis is sensitive to this antibiotic. And then we have it is present in normal microbiota of skin, as it is epidermidis, and it contaminates blood cultures. It infects prosthetic devices, example, hip implant, heart valve, and IV catheters by producing adherent biofilms. So producing biofilms is an important distinction point for Staph epidermidis. Then we have Staph saprophyticus, which is again gram-positive, catalase positive, but coagulase negative, and urease positive cocci in clusters. It is novobiocin resistant in comparison to Staph epidermidis, which is novobiocin sensitive. It is present in normal microbiota of female genital tract and perineum. And the second most common cause of uncomplicated UTI in young females, most common is E. coli. And this is an important point that it is the second most common cause of uncomplicated UTI in young females, most common is E. coli. Then we have Streptococcus species, Streptococcus pneumonia. It is gram-positive, but alpha hemolytic species. It is lancet-shaped diplococci. It is encapsulated. It has IgA protease. It is optogene sensitive and biosoluble. So we have to see what is optogene sensitive. It is a key lab test used to presumptively identify Streptococcus pneumonia, which is typically inhibited by optogen, unlike other alpha hemolytic streptococci. So it is ethyl hydrocuprin hydrochloride, a quinine derivative that selectively inhibits the growth of Streptococcus pneumoniae. So Streptococcus pneumoniae is the only one that is sensitive to optogen. And most common causes are MOPS, meningitis, otitis media in children, pneumonia, and sinusitis. Pneumococcal pneumonia is associated with rusty sputum. Patients with anatemic or functional hyposplenia or asplenia are predisposed to the infection because this is an encapsulated organism, and if the patients do not have a spleen, then it cannot be phagocytized. And there's no virulence without capsule. So if you remove the capsule, or if the vaccines target the capsule, then we can get rid of this organism. The pneumococcal vaccines are available in both conjugate PCV13, PCV15, PCV20, and polysaccharide formulations. The polysaccharide formulation is called PPSV, and the conjugates are called PCV13, PCV15, and PCV20. Again, polysaccharide, PPS, and conjugate PCV, 13, 15, 20, and 23. Then we have viridans groups to streptococci, which are also known as group D streptococci, I think. Yes. Pyogenes was group A, and A. was group B, and viridans is group D. It is gram-positive, alpha hemolytic cocci. It is optogen resistant in comparison to strep pneumoniae, and bile insoluble, again in comparison to strep pneumoniae. Normal microbiota of the oropharynx. It is present in normal microbiota of the oropharynx, and Streptococcus mutans and Streptococcus mitis cause dental caries. I think these are the strep mutants we have to see. Strep mutants. It is gram-positive bacterium that plays a central role in caries. And what is its relation to viridans? It is a member of the viridan streptococci group. So this can cause dental caries. And S. sanguinis, which is also a member of this group, makes dextrons that bind to fibrin platelet aggregates on damaged heart valves. causing infective endocarditis. So there's a damaged heart valve, and sanguinus is going to come and make dextrons on it, and make dextrons, and these dextrons would bind to fibrin-plated aggregates on damaged heart valves, and these dextrons will then cause infective endocarditis. Then we have this viridans group strep live in the mouth because they're not afraid of the chin. They're oxygen resistant. Sanguinus is equal to blood. It means blood. Think there's lots of blood in the heart. So sanguinus is for infective endocarditis. Mutans is for dental caries, and they are living in the mouth because they're not afraid of oxygen. Then we have strep pyogenes, group A streptococci. It is gram-positive cocci in chains. Group A strep causes pyogenic pharyngitis, cellulitis, and impetigo, which is honey-crusted lesions, and erysipelas. So, impetigo, first slide. Impetigo. Okay, this is a honey-crusted lesion right here, caused by our very strep pyogenes. Then we have erysipelas, or that is what it sounds like. And erysipelas looks like, okay, it is a red erythematous infection of the skin. And what is the difference between cellulitis and erysipelas? Cellulitis refers to acute superficial diffuse spreading skin infection, may refer to infection involving the deeper dermis and adjacent subcutaneous tissue. And erysipelas may refer to a more superficial infection. Cellulitis is deep and erysipelas is superficial. And pyogenic is causing pharyngitis, cellulitis, impetigo, and erysipelas. Toxigenic effects would be scarlet fever, toxic shock syndrome, and necrotizing fasciitis. If I search what necrotizing fasciitis is, it is a rare, rapid, progressive bacterial infection that destroys soft tissue and can be life-threatening, requiring immediate medical intervention. And if I look at a diagram of it, this is how this necrotizing fasciitis looks like, right? Immunologic would cause rheumatic fever and glomerulonephritis. It is bacitracin sensitive. And what might bacitracin may be? Bacitracin. It is a topical antibiotic used to prevent infections and minor cuts, scrapes, and burns by inhibiting material cell wall synthesis. And it is effective against gram-positive bacteria, such as strep pyogenes, which is bacitracin sensitive. And it is obviously causing pyogenic pharyngitis, cellulitis, and impetigo. It is bacitracin sensitive. It is beta hemolytic. It is pyrrolidonyl aryl amidase PYR positive. Hyaluronic acid capsule. This is a virulence factor. And M protein inhibit phagocytosis. M protein is also a very important factor in it. Antibodies to M protein enhance host defenses. Strictly similar to host proteins, that is myosin, can lead to autoimmunity, that is carditis seen in acute rheumatic fever. Diagnosed of pharyngitis by a throat swab, which can be tested with an antigen detection assay, rapid in office results, or cultured on blood agar results in 48 hours. Okay. So, we'll have to search a few things such as what is PYR positive test. Test is PYR test. Rapid biochemical assay used to detect this activity in bacteria, primarily for identifying Streptococcus pyogenes and Enterococcus species. Okay, so it is a test for this one. An absence of an enzyme results in no color or yellow color. That indicates a negative result. So, hyaluronic acid, M protein, and PIR positive, and also beta hemolytic and bacitracin sensitive. And strep pharyngitis can be detected by a swab that is tested with an antigen detection assay. Rapid results or cultured on blood agar that results in 48 hours. Pyogenes, pharyngitis can result in rheumatic fever and glomerulonephritis. Strains causing impetigo can induce glomerulonephritis. Key virulence factors include DNase, okay, erythrogenic exotoxin, streptokinase, streptolysin O, ASO titer, which is some antigen-specific streptolysin O titer, or anti-DNase B antibodies indicate recent strep pyogenes infection. And scarlet fever is something that is fine, blushing, generalized sandpaper-like rash sparing palms and soles, strawberry tongue, and sycamore pallid in the setting of a streptococcal pharyngitis, and it is erythrogenic toxin positive. So, a presentation of scarlet fever would be nice. Okay, so these are the three symptoms: sycamore pallid, strawberry-like rash, and the third one was rash sparing palms and soles, right? Then we have Streptococcus agalacta, which is group B streptococci. It is gram-positive cocci. It is specificity resistant. It is beta hemolytic and colonizes the vagina. It causes pneumonia, meningitis, and sepsis, mainly in babies. Polysaccharide capsule confers virulence, produces CMP factor, which enlarges the area of hemolysis formed by Staph aureus. CMP stands for the authors of the test, not cyclic AMP. Oh, so it is basically CAMP. You can call it CAMP factor. Lipirid test positive, PYR test negative, unlike Streptococcus pyogenes that has, sorry, yes, that has a positive PYR test. Then we have screen pregnant patients at 35 to 37 weeks of gestation with rectal and vaginal swabs. Patients with positive culture will receive intrapartum penicillin or ampicillin prophylaxis. And group B for babies. The second last one we are going to do is Streptococcus galoleticus. It is formerly called S. bovis. It is a gram-positive cocci that colonizes the gut, can cause bacteremia and infective endocarditis. Patients with S. galoleticus endocarditis have increased risk of colon cancer. So it is an important one. Bovis in the blood, cancer in the colon. Oh, that's such a nice mnemonic. So bovis in the blood can cause bacteremia and infective endocarditis and cancer in the colon. Yep. The last one is enterococci, which is a gram-positive cocci. Enterococci is Eficalis and Eficium, which are normal colonic microbiota that are intrinsically resistant to penicillin G and cause UTI, biliary tract infections, and infective endocarditis. Following GI or GU procedures, they are catalase negative, they are PYR positive, and they're typically non-hemolytic. Vancomycin resistant enterococci are an important cause of healthcare associated infection. Enterococci are more resilient than, wait, I misread that. Vancomycin resistant enterococci are important causes of healthcare associated infection. Okay, so they are resistant to vancomycin and this they're important for healthcare associated infection. Enterococci are more resilient than streptococci and they can grow in 6.5% NaCl and bile. Entero means intestine, fecalis means feces, strepto means twisted, and coccus means berry. Yeah, so that was about it. It can grow in 6.5% NaCl and it is intrinsically resistant to penicillin, non-hemolytic, and vancomycin resistant enterococci. So this was it for this one. We'll do the gram-negative bacteria in our next video. See you then. Bye.